So
So one
let
more thing I want to
yeah, tell me.
like I have a class at 8 p.m. That
Mhm.
is 10:30 Indian timing. So is it fine if we like just take it for 1.5 hour because I have to attend that. It's regarding practicals only.
Okay, today. Yeah,
Yeah.
it's fine. It's up to you. No worries. Okay. So let's quickly start with some important things, if you remember. So in this chapter, we studied about some both facts and also concepts. Some experiments were important part of this chapter, like this Griffith's transforming principle. This is often asked in boards, inevitably, and all the experiments that followed to prove that DNA is the genetic material. So for boards point of view, you have to be thorough with what were the experiments and in what series these experiments were being done to prove that it is indeed DNA, which is the genetic material. Yeah. So I hope the series is clear to you, starting with Griffith's transforming principle. And I'll just quickly go through things. Stop me wherever you feel you have any difficulty in understanding. And it's like I'm kind of giving a quick recap so that you are on the same page before we do some questions. Okay. So
Okay.
it all started with Griffith's transforming principle, where Griffith, while doing some experiments with Streptococcus pneumoniae, he realized that out of the two strains, which is called S strain, the name S is for smooth, and the name is derived from the mucus shiny colonies that they produce. So the smooth strain or the S strain, which was lethal upon infection, and there was another strain called R strain, which is rough, which formed rough colonies, non-shiny, and that was not lethal. So infection with the R strain will not lead to death, but infection with the S strain will lead to pneumonia, development of pneumonia in the lungs, and finally the organism will die. So he did this experiment where he injected S strain and R strain single, and he realized that, as people knew, S strain killed the mice, but R strain did not. But then he heat killed the S strain, and heat killing is basically making the bacteria unable to cause infection and unable to replicate, which is like you weaken the bacteria so that the immune system of the organism, the moment the bacteria will enter in the body, immune system will be more powerful and will get rid of the bacteria. So the immune system could win over this heat kill S strain, and then the mice lives. But interesting thing was when he mixed a heat killed S strain, which was unable to kill the mice, with the live R strain, which was again unable to kill the mice. This time the mice died. Now there were many questions that arise from this, whether the heat killing was proper or not. He did all those repetitions again and finally concluded that indeed there is something that gets transferred from the S strain, even though it is heat killed, to the R strain, which is live, to convert the R strain into S strain. So this property of virulency can only be transferred through something as powerful as genetic material in the cell. So he said that this is what is genetic material, and it allows transformation of R strain into S strain. So he gave this transforming principle, and he said that genetic material is responsible for it, but still it was not known what is genetic material and how it is doing this transformation. So Avery, MacLeod, and McCarthy, this trio, then uh, took this experiment forward in their own ways and intelligence. They thought of purifying the chemicals from the heat killed S strain, like all the chemicals. Now, purifying all the chemicals is a very tedious task because inside a cell, you have everything: proteins, lipids, sugars, nucleic acids, both DNA and RNA. And it was not known which among these is a genetic material. So they isolated all, and then um, With the help of digesting enzyme, they degraded one after the other and looked at which of the following is responsible for causing the transformation. Though basically it was a very good experiment, theoretically and logically very sound, but people started, they didn't expect, they didn't accept the results as conclusive proofs uh, because of concerns and questions that maybe this experiment was not done as cleanly as it should be. There could be some contaminations, so it is not enough to prove that what is a genetic material. They found that in case of where you where you denature nucleic acids, the transformation did not happen. Okay, but then Hershey and Chase, Alfred Hershey and Martha Chase, they provided an unequivocal proof, which was also a visual proof that DNA is the genetic material and not protein. And so so by this time, long story short, between 1943 to 1952, almost 10 years, in this time, people kind of converged to either protein or DNA being the candidates. So they did an experiment where basically they use radioactive labeling. In one case, they labeled virus, the virus protein part, and in another case, they labeled the virus's genetic material part, which basically they labeled the DNA, but now we know it's the genetic material. And they allowed these viruses, the bacteriophages, to infect E. coli like they do. And then after the infection, they they did centrifugation and separated the viral particles from the bacteria. And by this time, if infection stage has happened properly, the virus must have been successfully um, sent the genetic material inside the bacteria. So upon centrifugation, as the bacterial particles settle at the bottom, they realized that you could only see radioactivity in the case where they have labeled DNA, but not in the case like radioactivity in the sediments. So sediment is the place where bacterial cells are because bacterial cells will be heavier than viral particles. And when you will centrifuge, they will settle at the bottom, like, right? Because they are big cells. So if in the sediment you see radioactivity, which means whatever was the genetic material is in the cells now, in the bacterial cells now. In case of protein labeling, they saw that the radioactivity signal is coming from the supernatant, which means protein didn't enter the bacteria. But in case of DNA, they saw that the radioactive signal is coming from the sediment, which means DNA did enter the bacteria. That's why it's also get precipitated with the bacteria. And this proved that genetic material is DNA because this is what entered the bacteria and not the protein. Okay, and it's known that a virus only sends its genetic material inside, a bacteriophage only sends its genetic material inside, and that's how it replicates. So this was a proof that DNA is the genetic material. So all these experiments in series, I hope is crystal clear, right? You know,
Yeah.
yeah, cool. Now also remember about RNA world hypothesis and what are the proof that supports RNA world hypothesis. So mostly they ask about the proofs that support that indeed once there was a RNA world, which then led to you evolution of DNA. So we have certain proofs like we know that RNA has the capability of both uh, having storing the information and also it can catalyze very important reactions. In fact, still there are some biochemical reactions in the body which are only catalyzed by enzymes which are RNA. So they are called ribozymes. Now, the presence of ribozymes is a proof that RNA once was able to do both, catalyze the reactions, which is very important because if you cannot do that, you cannot do metabolism. So remember that what is important for any cell to be a living system, right? The three basic necessities. of the most primitive life is it should have a boundary, which is made up of lipids. We know that it should have a molecule that can store information and can replicate. Now, to replicate, that molecule needs some other molecule's help. Now, enzymes were not there in the beginning because enzymes were protein. Now, and proteins come at the very last stage of central dogma. So you need a single molecule that could do both, store information and catalyze reactions. And that was RNA and not DNA. DNA does not have the capability to catalyze reactions. It is a very good storing molecule. Okay. So remember these things. And with this, any doubts? You know?
No.
Okay, cool. Let's see. Then we talked about the three processes of central dogma: replication, transcription, and translation. And don't get confused with enzymes of each process. Easier way to remember is you have to see what is being formed and keeping what as a template. So a replication is copying of a DNA into another DNA, right? Making DNA from DNA. So the enzyme that will be responsible will be a DNA dependent enzyme. So DNA dependent DNA polymerase is what does the replication, right? And we know that replication is semi-conservative. And for that, the proof came from Messelson and Stahl's experiment, where they used heavy nitrogen, not radioactive. Okay. Remember that there can be questions to confuse you where radioactivity will be mentioned to prove the semi-conservative nature of DNA replication, but it was not radioactive DNA. They used the Messelson and Stahl used a heavy DNA using nitrogen 15. So this experiment also is uh, quite brilliant and straightforward. They cultured E. coli in heavy nitrogen for many generations, and once they did that, it was to make sure that all the DNA in all these bacteria will be carrying heavy nitrogen. And then they allowed it to grow on normal nitrogen. And after one generation, if the replication was fully conservative, which means each DNA the parental strand remains parental and a new progeny strand is formed. In that case, you will get one full DNA with heavy nitrogen and another full DNA. By full DNA, I mean both the strands of DNA with uh, normal nitrogen, but that did not happen. Instead, what they got is after one generation, they got a mixed band, which means it was neither heavy nor light. It was some hybrid. And this proved that one chain was heavy and one chain was light. So one chain came from the parental strand, which was heavy, and one chain was the newly formed, which was light in the nitrogen 14 environment. Correct? And if you'll go to the next generation, again, you will see that there is no uh, amplification or increase in the heavy strand. The heavy strand stays only, there was no heavy strand, like complete double heavy strand. There were two hybrid strands, and almost all other strands which were made were light strands. So this proves that DNA replicates in a semi-conservative manner, which means, in simple words, that the two strands of the DNA of the double helix open up, and each parental strand then becomes a template for the new strand to be formed around it. Correct? You understand, Aaron? So this is
Yeah.
this is semi-conservative method. And the similar kind of experiments were then done and proven by Dr. Taylor, and he used radioactive thymidine. So remember, Messelson and Stahl used just heavy nitrogen, and you can do a similar kind of experiment. He did it in faba beans using radioactive thymidine. Then the replication machinery, just remember these enzymes name and their function. These are often asked. And more importantly, what is leading strand and what is lagging strand? If you have some clarity on leading strand and lagging strand and why are they called so? You know, what is a
Um,
leading strand and what is a lagging strand? You can take the help of the image here on the board. So this is a picture, diagrammatic view, cartoon view of replication fork, and you can see two words, lagging strand and leading strand. What does this mean?
um, a leading strand is synthesized by adding nucleotides to 3' end, and
But why? Yeah, and the lagging strand,
the lagging strand is synthesized continuously.
right? And why is that? Why is lagging strand is lagging strand synthesized continuously or leading strand synthesized continuously? So once you're right, one strand is continuous and the other is discontinuous, right?
Yeah.
Which one is continuous and which one is discontinuous?
The one which is like 5' to 3', that is, um,
So both the strands will have one so both the strands, when they will replicate, they will have a 5' to 3' end. But taking parental strand as the template here, you are saying when the parental strand is 3' to 5' or 5' to 3', which one is leading and why it is called leading? So you know the direction in which the polymerase enzyme works, whether it is a DNA polymerase, a
5'
DNA
to
dependent
3,
DNA, right? It works in
5' to 3' direction.
5' to 3' direction. Okay. So basically, in the case of a lagging strand, it's called lagging because if you see the direction in which DNA polymerase works is the direction of the template strand. So DNA polymerase cannot continuously work because the complementary strand that it is making, it has to make it in 3' to 5' direction, opposite to its own direction of work. But since polymerase cannot move in a 3' to 5' direction, it waits for opening of this helix. And the moment it opens a little, it synthesizes the complementary strand. As you can see here, can you see my cursor? You know,
Yes.
so this part got synthesized and you can see there's a break here because then again, some more part got opened and again the enzyme will come and sit here and synthesize in 5' to 3' direction. So this is how it will keep synthesizing the complementary strand in bits,
Hmm.
in fragments. So the lagging strand is called lagging strand because it is not as fast and continuous. So it lags. And there are fragments that are formed, and these fragments are called Okazaki fragments. So at the
Yeah.
lagging strand, all these Okazaki fragments have to be joined together. So there is one more enzyme that comes into picture, which is called this blue thing that you see here. Can you see my cursor?
Yes, sir.
You will see this enzyme, which is called DNA ligase, working on the lagging strand to join the pieces together, but it will not be working on the leading strand because the leading strand continuously gets synthesized. And why so? Because the template strand of the leading strand is in 3' to 5' direction. So the complementary strand has to be made in 5 prime to 3 prime direction, which happens to be the direction of RNA polymerase, sorry, DNA polymerase is working as well. So simple. So it's very straightforward. RNA polymerase can just, sorry, DNA polymerase can just sit at one end and continuously keep going. It will sit here and continuously keep synthesizing as the replication fork will open up. Okay. But on the other hand, on the opposite strand, it is discontinuous in fragments. So the discontinuous strand is the lagging strand, and the continuous strand is the leading strand. Is it clear?
Yes, sir.
Yeah. In other words, if we talk about directionality, keeping the parental DNA in mind, the 5 prime to 3 prime strand of the parental DNA will be producing the lagging strand, right? So you can see here 5 prime to 3 prime. This one is the lagging strand, and the 3 prime to 5 prime of the parental strand will be the leading. And it makes sense because the complementary will be then 5 prime to 3 prime, which is the direction of RNA polymerase, right?
Yes.
Okay, cool. So remember the replication fork, and then transcription is copying information of DNA into RNA. So it's DNA dependent, RNA polymerase, direction remains the same. And here, the rule of complementarity is followed. Just remember that there is one change here, which is RNA does not have RNA does not have thymine, but it has uracil. So if you are making a RNA from DNA, all the information that's coded for thymine by a DNA gets converted into uracil when you make a RNA. Okay, cool. Then transcription unit, just brush it up. We'll do some questions on this part, so it will get cleared with some questions. And then in prokaryotes and eukaryotes. So what is the difference between transcription in prokaryotes versus transcription in eukaryotes? If I ask you one, the most important difference.
Uh, in eukaryotes, it takes place inside the nucleus, and in
Mhm.
prokaryotes, it takes place inside the cytoplasm.
But that's because they don't have a nucleus. That's not a difference between transcription. That is the difference in where it happens. You understand? I'm asking about the process. So that's where you have to understand the question. So if you write this, the one who's checking can just not give you marks, and you cannot contest, because if bacteria had a nucleus, then it would have happened in the nucleus there also. So it's about the location, but I'm asking about the process itself. Is there any difference in the process regardless where it is happening? It's transcription that is happening. So what is the difference in the transcription of a prokaryote versus the transcription of a eukaryote? Now, if you remember, we talked about um, how is the gene structured in prokaryote and eukaryote. Do you remember that?
Yeah, yeah. So
Yeah.
in like
Mhm.
so in I think in eukaryotes we have split genes that is continuous, whereas in you no in prokaryotes we have non-split genes.
Mhm.
Wait, wait. Yeah, prokaryotes have non-split genes.
So there was a term for it, monocystronic and polycystronic.
Yeah,
So which one
monocyst.
has? Mhm. Is a prokaryote monocistronic or polycistronic? That will help you understand basically translation well. But since you brought up that topic, that also makes some connection here. So prokaryotes are polycistronic, right?
Yes.
Which means within the same promoter, they have
So your voice is
multiple
breaking.
genes which contained many same regions. Okay, just one second. Is it better now, my voice?
Yeah.
I'll just wait for a few seconds. It stabilizes sometime. Is it better? You know?
Yes, sir, it's better.
Okay, good. Sorry, it's the fluctuations. So we talked about the transcription unit, right? For both prokaryotes and eukaryotes. And I told you that the split gene arrangement is present in the eukaryotes. When I say split gene, it simply means that each gene will have certain splits of exons and introns. Exons and introns. Exons are the region that are important, and how to remember it is by EX. It exists in the actual gene, and introns are the regions that are not required. Okay. So this split gene arrangement, which is present in eukaryotes, also protects eukaryotes from many mutations that happen. Why? Because if a mutation is happening in the region where a gene is, again 50% chances are that it can happen in introns. So you are saved because introns ultimately will be removed. Yes or no? And your exons will exist in the actual gene, and you get some benefit. Now, we don't know if this is the reason why it evolved like such, but this is how it gives us benefit. So in humans, it is monocistronic, or in eukaryotes, it is monocistronic. And cistron is the segment of DNA that codes for a protein. Remember, I told you that all genes that make RNA finally do not end up making protein. Remember that discussion?
Yes, sir.
All the And what are examples of it? Any example where you have a gene that gets transcribed into RNA but does not gets translated into protein? Very simple. What are the type of RNAs that are functional in the form of RNA? Remember rRNA, eRNA, correct? So
Yes.
tRNA is functional as tRNA. It is functional in its RNA form. It does not need to produce protein. So the gene encoding for tRNA, rRNAs, there are called long non-coding RNAs. So all these, they get transcribed into RNA but does not further go on to form proteins. So the field needed more clarity on this. So a new term was invented called cistron. Now, if you call a segment of DNA as cistron, then you can be sure that this piece of DNA or this segment of DNA will for sure give rise to protein. So humans and eukaryotes are monocistronic, which means there is a single protein coding sequence between the promoter and the terminator, okay, in its unit, transcription unit. But in case of prokaryotes, it's called polycistronic. There are multiple protein coding units between a promoter and a terminator, and they're all sandwiched together. There are multiple genes, and each can give rise to different protein. Okay, that's why in case of prokaryotes, because there is no split gene arrangement, there is no introns and exons, everything is exons only. So if everything is water, you will not call it water and something else which is not present. You will just call it water. So here everything is exon. So when transcription happens, you don't need to remove anything from the um, resultant mRNA. But in case of eukaryotes, when an mRNA is formed because of transcription, the mRNA contains exons, introns, exons, introns. And you need to remove introns and stitch back all the exons together. And this process is called post-transcriptional modification, right? So post-transcriptional modification is one important factor, one important difference between prokaryotic and eukaryotic transcription. A prokaryotic transcription produces fully functional RNA right at the beginning. And the proof is that in a bacteria, the moment a RNA starts to be produced, suppose 10% or 20% of the mRNA is synthesized, at the same time, it will also start getting translated. So ribosomes will come and sit and will start processing it because it's a complete, it's a, what do you say? It's functional mRNA anyways. Do you understand that?
Yeah.
But in case of eukaryotes, even when the mRNA is fully synthesized, first there is a capping and tailing that happens to protect the mRNA. Then there is splicing that happens to remove the introns. And finally, the mature mRNA is ready. And then this mature mRNA, which is called mRNA, messenger RNA, then it leaves the nucleus and goes into the cytoplasm. And then the translation starts. So long journey. Okay. So post-transcriptional processing is very important, and it is the difference between the two. Okay, cool. So let's go and do some questions. So I will stop sharing this thing here, and let's start with something along these lines. A question that has come in board also. Can you see the question, Iram?
Yes, sir.
Question 175. Yeah, clearly, can you see it? Uh, so there are three parts, A, B, and C. And without taking the help of the book, because this question is directly from NCRT, just take a look, take 2 minutes, and see if you can answer A, B, C all. Okay, don't look at the books. Uh-oh, what happened? Yeah, the time starts now.
So I've answered in the chat.
Okay. Yes, so first is the repressor, and how does this molecule um, gets inactivated? The hint is again given in the diagram itself. How does it get inactivated?
When there is the presence of, no, when lactose is absent.
When lactose is absent. So lactose is normally absent. That's where the you know this lac operon is shut off. When lactose is present, because lactose is the inducer. So they have written the word inducer here. So you have to write that in the presence of inducer, which is lactose here, because this is lac operon, it comes and binds to the repressor and inactivates the repressor. So basically you are inactivating, you are repressing the repressor. Make sense?
Yes.
So when you repress the repressor can no longer go and bind to the promoter region, which is written, which is denoted by P here. So this P is from where this whole polycistronic gene is transcribed. So if repressor, which is X, sits on P, then polymerase cannot sit because the space has already been occupied. But if the repressor is repressed by lactose, then polymerase is free to come and sit on the promoter and start the transcription. Okay. Remember the inducer here is lactose. Okay. You know? So in the
Yeah.
presence of lactose, the repressor gets inactivated. Make sense?
Yeah.
Perfect. Yeah. Second part. So you're right. With the second part, you said which one of the structural gene codes for beta galactosidase? So you said Z. Yes, that's correct. And also, what about the others? Y and A. What does Y codes for?
Permease
Mhm.
and yeah, permease.
Permease and
I'm not
beta
able to recall.
galactosidase, permease, and
Permease, permease, trans, mm,
transacetylase.
transacetylase. Yeah.
Yeah. Okay, perfect. Cool. Even if you remember this much, it's fine for your boards. But for entrances, you need very clear picture in your brain. Cool. Excuse me. See, when will the transcription of this gene stop? When do you think transcription will stop? You're right in a way that you said lactose is absent. Yeah, you have to just elaborate it a bit. You have to say that when lactose, which is the inducer, is no longer present in the surrounding, then it can no longer repress or inactivate the repressor. And then the repressor will sit on the promoter and will not allow polymerase to sit, and this transcription of this gene will stop. Make sense?
Yes.
Perfect. So this is the kind of questions they ask in the boards. I would also like you to solve some questions. And since you are only one, so you can speak up, okay? So just speak the answer. Let me share some, wait, some multiple choice questions, okay? We'll do some MCQs. Um, just give me one moment, okay? Yeah. Yes. Can you see this particular question number 20 here? You know,
Yes, sir.
yeah. So can you read the question, and then when you're sure, just say that which is the right answer and why do you think so? So you just don't have to tell the right answer, you have to justify it also. So that's the part of your practice. Read the question. You so you have to read it so that I can also hear. If you are okay. Are you there, Iram?
And
Yeah,
yes, find
yeah,
out the wrong statement about heterochromatin. It is densely packed, it stains dark, it is transcriptionally active, it is late replicating.
yeah. What is wrong? Which statement is wrong? Remember, focus on the word wrong, okay? Remember that. Sometimes they give you, so these kind of questions are very common. They will ask you a question and it will be like, which of the following is not an arthropod? So if you don't follow on the word not, you may end up choosing the wrong option. Yes.
It is transcriptionally active.
No, that's not wrong. That's right.
Hmm?
Heterochromatins, right? Find out the wrong. So you have to find out the wrong statement. No, you're right, actually. It is transcriptionally
Yeah,
inactive,
it
right?
is. Yes, that's
Yeah,
what.
yeah, sorry. Got confused. Yeah. So heterochromatins are transcriptionally inactive. So C is the right option because it is the wrong statement about heterochromatins. And the
Yeah,
last one, it is late replicating. What does it mean? See, this is not addressed in your NCRT.
yeah,
So that's
exactly.
that's where, yeah, that's where I say that mostly when people teach, make students prepare them for NEET, they say that don't worry, everything will come from NCRT. I say no. Sometimes concepts are in NCRT, but explanations or concept building of those concepts you will not find it in NCRT. So there was nothing about replication of euchromatins or heterochromatins or any difference between the two in NCRT. But it's logical. See, what is heterochromatin? Why it stains dark? Because it's densely packed. And what do we mean by it's densely packed? There are proteins that keep this part of the DNA in a chromosome very, very, um, what do you say, supercoiled. And this is the reason that it is not transcriptionally active because the molecules which are responsible for transcription cannot reach there. And if that is true, the same molecules which are responsible for replication will also find it difficult to reach here, right? It won't be very easy for all the protein machinery to reach this part of the DNA. Correct? Make sense? Yeah.
Yeah,
So just by logic, you can understand that if a DNA is being replicated, first to replicate, it has to open up, uncoil itself. That's what the enzyme helicase was doing, right? Topoisomerase. It will unwind the helical form of DNA, open it so that replication fork can be made. Since it is very, very densely packed region, it will take some time to open this. It's like a clenched fist. If you have to, you know, take something from an open fist versus a clenched fist, it will take a lot of time in a clenched fist because you will first open it. So that's why it is late replicating. These regions replicate in the last. And that's where I also told you replication does not start from one end and goes all the way to the other end of the DNA. There are pockets, right? Where the DNA opens, it replicates, and they keep moving. So these regions are called origin of replication. We covered that part in biotechnology. Remember that?
Yes, sir.
In a normal DNA also, there are multiple origin of replication sites. So a heterochromatin can never be a origin of replication. Correct? Again, this is not addressed in NCERT, but this is an exact neat question. Which of the following sites in DNA cannot serve as origin of replication? And if they say promoter, adjacent to promoter, enhancers, and all these words, one of the word will be a heterochromatin. So heterochromatin can never be origin of replication because it's tightly, densely packed. It needs to open first. Make sense, Iram?
Yes, sir. So one more
No.
thing.
Yeah.
So where do this exam setter get these extra questions from?
They don't. So yeah, I don't know answer to this question. Where do they get so these questions are just based on the concepts that are taught to you, right? Earlier, when the syllabus was not just limited to NCRT, there were many exams, right? There were different state exams. There was All India. It was called at that during my time. Um, it was known as AIPMT, All India Pre-Medical Test. And then there were different state exams as well. They all merged it and call it NEET, National and Eligibility Entrance Test. Then they had to standardize the syllabus also. But earlier they were like as deep as you want to go and study. So I remember that AIMS as an institution used to have its own entrance, and that was one of the uh, most difficult because they will ask you question which you will never find in NCERT anywhere. But the concept will be there, either just you know slightly discussed or discussed in detail, but the concept will be there. So NCERT is a good NCERT is actually a heterochromatin served to you. Okay, information is there, but densely packed. So a lot of things in just one sentence, like that. So they are all derived from the same concept. So since you know that heterochromatins are densely packed, one of the question could be that it is transcriptionally inactive or active. Other could be on the basis of whether it will be replicating faster or it will be replicating slower. So of course it will replicate slow. So they are late replicating sites. Then on the same concept, they can ask you, can they be origin of replication sites? No, because they replicate late. So they cannot be origin of replication sites. Okay, make sense?
Yes.
Cool. Okay, let's also there's some more questions I have thought out. Um, please, yes, let's go for replication part. But this, yeah, can you read this question 31 and tell me which option you will choose? Question 31.
Sir, option C.
Option C. Why? You have to justify also. Option C
The
is
uh,
so the question is which of the following statements are incorrect? And you are saying 1, 2, and 3 all are incorrect.
no. 2 and 3. Option B.
Why did you say option C first? So you are saying that option 2 and option 3 is incorrect. So RNA is more stable than DNA is incorrect. Okay.
Yes.
And RNA has evolved from DNA is incorrect.
Incorrect. Yeah.
Okay. Okay. And RNA acts as a genetic material as well as a catalyst is correct, right?
Yeah. Yeah.
Yeah, right. So it's 2 and 3, which is option B, right?
B. Hmm.
Perfect. So why did you choose C first?
So actually, I didn't think of retrovirus, so I was thinking of humans.
Oh, okay. No, even in retrovirus, RNA does not evolve from DNA, no? RNA is RNA can be in retrovirus, DNA can be synthesized from RNA, right?
No, no, no. The first one, right? First one says that RNA acts as a genetic material. So I was thinking for
Oh,
humans, but
okay.
it's
Yeah, no,
for
there are, yes. Right. Cool. So these type of questions. So don't be in haste while you're doing MCQs because the options are quite similar. Okay. Um, this you know easily. Don't get mm, you know, don't be scared by these big words. The first experimental proof for semi-conservative DNA replication was shown in which experiment they are talking about? Giving you no clue about the name, the scientist, or anyone. So you might feel a little.
uh, X. The B one is stereocola.
Exactly. And whose experiment they are talking about? We just talked about it.
Michelson and stuff.
Perfect. Perfect. Yeah, you got it. Okay. So I have cool. Question 35. So this is what you have to remember for both for entrances. Maybe boards won't ask you on specific enzymes.
All of these?
Yes, that's correct. Very simple. So you took some time out of caution, right? Just to be
Yes, sir.
100% sure. It's fine. You have, so see, um, let me tell you one thing about entrances, regardless of which entrance it is. It's equally about managing the time as it is about how much you know, your knowledge. Okay. So someone who knows more can also do, you know, very bad in an entrance if that person does not manage his time. And someone who does not know about 100% of the syllabus can also do reasonably well or better because of time management. Okay. So there will be questions where you will know the answer straightforward. And even if you know, make it sure twice or thrice, it will not take more than 30 seconds. So you know the answer, just take it. So you outright saved 30 seconds in that question, which you can use for the next one. But if the next question is also easy, then you saved more time and keep going for the next and the next, right? So you should also have a presence of mind, and this can only come when you have practiced enough at your home, at your by yourself, that stay calm and composed while you're giving an entrance. Do not get into the rush of an entrance because then you will not be able to take wise judgments that okay, I have saved some time in last 5 questions, so I need not, I can spend some time to understand this, or I will come back to this question once I go through the paper. So there are many ways to give an entrance. And what I mostly used to do is I will s- I will I will have it in three rounds. The first round, I call it the sweeping round. You just sweep through all the questions. Read the question, and if there is anything you understand, just look at the options. And if you know the answer, take it. Go to the next question. Read the question. If there is anything that you are doubtful about and you don't understand, skip it, go to the next. So it's like one round of sweeping. And in that round, let's say out of 100 questions, if you were able to do 30 or 35, that's good because you took very less time. You were just sweeping through questions and you did 30, 35. So these were the easier ones that you did. Those marks are secured. Then you again start from the beginning. And this time you will spend 1 minute on each question. Okay. And try to think if you can get to an answer or not. You understand? You know? So one sweep
Yes.
through will give you some confidence because you will have a idea of how many questions you already have in your pocket. Okay, does it make sense?
Yes, sir.
Okay, so yeah, this is the question I want to do on leading and lagging strand. Um, Question number 40. Can you see the whole question? Yes, right, with all the options?
Yes, sir.
Yes, read question for now. See, these kind of questions take time to read also, right? So when you see these questions, don't just leave it. Just give it a read because sometimes they are very easy and straightforward. So do this. Question 40. What do you think?
Sir, I think second is wrong. And
Why second wrong? First, give me an option. A, B, C, or D. Options are at the bottom. Which option you will take if this is an entrance?
A.
Option A. Okay. And why? Now so your time stops here. So you've chosen option A. Now let's see if you have got 1 mark or you have got deducted, no? Minus 0.25. Cool. So option A, 2 and 4. So why do you think 2 and 4 are incorrect?
Because
Differences.
in leading stand it is fast growth, whereas in the lagging one it is slow growth. So it's wrong.
Right. So 2 is of course incorrect. And that is the reason. See, 2 is the most obvious incorrect. And that's why, look, you will see 2 in every option. A, B, C, D all have 2 in it, right? So that's what make entrance a little tricky. Just looking at 2, you cannot decide because there is 2 in all. So you have to be sure about one more option. And you chose 4. Now let's read 4. Formation of leading strand begins immediately at the beginning of replication, and formation of lagging strands begin a bit later than that of leading strand. So what's wrong in it? I think that's true, no? Because leading strand, you yourself said that it is faster. So if it is faster, it just starts. It does not wait for anything. Whereas lagging strand is slower because it waits for the replication fork to open, a few like some distance, and then it will make a fragment. Then again it will wait for it to open, then it will make another fragment, and then these two will be called Okazaki fragments. That's why it's lagging, right?
So I chose this because I feel first and third options are correct. So
Okay, so you think first is obvious correct, right? That's why it is not there in 3 out of 4. Leading strand does not require ligase because ligase is to paste or stick back fragments. So in leading strand, there is no fragment. So it does not require ligase, like I was telling you, right? Through that cartoon depiction.
yes.
Lagging strand requires ligase to join Okazaki fragments. Makes sense. Outright correct difference. So that's why you will see that 3 out of 4 options does not have one. The option 3, if you see option 3, excuse me, it talks about the template. So about leading strand, it says its template opens in 5 prime to 3 prime direction. If the template opens in 5 prime to 3 prime direction, then the newly forming strand have to be in 3 prime to 5 prime direction. And if that is the case, it cannot be the leading strand because 3 prime to 5 prime is not the direction of polymerase. Don't you think so?
So I didn't get this one.
Yeah. So what is a template strand? Template is the parental, right? When the two
Yes, sir.
strands open so that DNA can replicate in a semi-conservative manner, one strand comes from the parent, right? And the other one is synthesized. So the strand
Oh
that
yeah.
comes from the parent is called the template. So if you are making a leading strand, your template has to be 3 prime to 5 prime so that the complementary strand will be 5 prime to 3 prime. And 5 prime to 3 prime is the direction of DNA polymerase. So it will be continuously formed. So option 3 is incorrect difference. It should be switched. So leading strand, you can just write it down. The template of the leading strand opens in 3 prime to 5 prime direction. And the template of the lagging strand opens in 5 prime to 3 prime direction. Do you understand it now, Iru?
Yes.
Yeah. So this is what you have to be careful about while you are solving questions in the entrance. Okay. So correct option is option C. Yeah. Okay. Perfect. Now there's one more question I want you to see. Yeah. These kind of questions, question 45, I will suggest you if this kind of question comes, do not do it, just by gazing an eye. Write it down. It might take 45 seconds, but don't take the risk. Okay. Write it down and it's simple. The, can you read the question 45 and give me the answer? Which of the option is true?
What would be the base sequence of RNA transcript obtained from given DNA segments?
Yeah. So the DNA segment is there are two strands, coding and non-coding.
Hmm.
What will be the RNA transcript?
Coding.
So coding strand is called coding strand because it provides the code for the RNA, right?
Yes.
Yeah. So, and whatever is the sequence of the coding strand will be the sequence of the RNA.
RNA transcript.
Yes. In your NCRT, the term coding and non-coding is not there. Instead, you know what is the term? Template encoding, right? If you go back to the figure, they say that one strand is template strand and the other one is coding strand. And remember, I told you during the class that coding strand is just called coding strand because the code is same as of the RNA, but it does not code for anything. It is the non-coding strand which acts as a template so that the RNA can be produced. Right?
Yes.
Yeah. So what will be the right option? A, B, C, or D?
A.
Yes, A. Perfect. Okay. Now, this is very simple. So questions like this also come, which is directly taken from NCRT, question 53. Refer to the diagram. What does it represent? The moment you see intron, exon, it can't be transcription in prokaryotes, right? And
Yes.
it
It's
is also
eukaryotic.
not it is also not translation because protein is not being formed here. So it is option B, transcription in eukaryotes, correct? Very simple. Anyways, but there are some difficult questions that will come to, one of which is, um, yes, this one. Um, Question 78, concept based. Can you can you read just read it and tell me what do you think?
Refer the given mRNA segment. It can be
Mhm.
translated completely into a polypeptide. Which of the following codons may correspond with A and B? AUG is the initiator, and terminator is A U A G U A G U G A. Option wait. So this is so confusing. Like they've given two options for A.
Right. And NCRT does not talk about any other codon that can act as start codon, right?
Yes, initiator is just AUG.
Yes. So AUG is initiator, but if you have to choose anything apart from AUG that can still make translation happen, then it's GUG. Okay, AUG and GUG. So option A, you see AUG and GUG. So this is not in NCRT again. I was just giving you an example, but B is there in NCRT, right? All the three stop codons, UAA, UAG, and UGA is there in
Yes.
NCRT. Okay? So remember, but see, option B and option C cannot be the right option, right? So here, even if you don't know that GUG can also act as a start codon, look at other options. In B, For A, the site A, they have given UAA and UGA. Both of them are stop codon, right? So they cannot act as A. They
Yes.
cannot be at the site A, right? So option B is outright rejected. It is very, very wrong. Option C, since AUG is there, but the next one is UGA, which is a stop codon. At A, you cannot have a stop codon,
Yeah.
correct?
Yes, sir. C is
Yeah.
option.
And in D, yeah, in D,
U U
you don't
U
know about GAG.
doesn't it?
Yeah, you don't know about GAG, but see, there is a UUU in stop codon. So UUU does not act as a stop codon. It is phenylalanine, I think, right? I think so. I can be wrong.
Yes, it's
Just check
phenyl.
it. Yeah. So it's phenylalanine. So that cannot be here. If it is here, then it will not be translated completely into a polypeptide. It will continue forward. So by just negating B, C, and D, you will get to know that, okay, A is the right option. See, even if you don't know GUG, right? And remember I told you that when we teach biology at junior classes, uh, we first
So,
talk about everything which is standard. Yes, right? You have a
so
question?
about like GUG, so in principle of inheritance, we studied about sickle cell anemia in which GAG turns to GUG.
Hmm.
That was a mutant codon.
Exactly,
Does it
but
make
that was
sense?
yeah, but that was exactly at the sixth position of the hemoglobin beta chain. So GUG is present millions and trillions of times in everyone's genome. It might be present in every gene also, but that's not what the point is. The point is if a GAG is present at the sixth position, which it is present in the beta chain of hemoglobin, and if that gets mutated to GUG, that causes sickle cell anemia. Not any GUG. Here they are talking about the first codon.
Yeah.
And
Okay.
this is just a random mRNA segment. So it's not like anywhere if GAG mutates to GUG, it will cause sickle cell. No. Even in the beta chain, in the gene of the hemoglobin beta chain, if there is a GAG at the 148th position, if that gets mutated, there won't be a sickle cell anemia, right? But exactly at the sixth position that mutation happens, it becomes sickle cell anemia. Why? Because that locus, that position is very important for proper shape. Okay,
Yes, sir.
cool. Okay, so 78 was, that's why I wanted you to do it. I'll take few more questions here, which are important. Um, this is from lack of rules, regulation. Yes. Question 99. This kind of question be ready for from not just from DNA fingerprinting, but from even biotechnology. So process-based question where they talk about steps. So you should know the process crystal clear so that you can say which step comes after what. They can make a similar question for gene splicing or transcription in eukaryotes or gel electrophoresis process in biotechnology or making a competent cell through heat shock and divalent cation therapy, anything.
Option B.
Can you?
Yes, sir.
Yeah, yeah. Read question 99.
So option B.
Option B,
Yes.
which is, so again, logic is isolation of DNA has to be the first thing, correct? So that's why you see isolation of DNA 4 and 4 in option A and B, right? It has to start from isolation of DNA. The question is about arrange the various steps of DNA fingerprinting technique in correct order. So you, which option you chose? Sorry, I forgot.
Option A.
Oh, I don't think you said option A in the first place.
No,
I think
I,
you
I
said
told
B or.
option B, but now I think it's A.
Okay, great. So, which one you want me to lock? Full and final.
A.
A. Okay. So you still start with 4, which is isolation of DNA. Once you isolate DNA, what will you do? The question is two or one. So two is digestion of DNA by restriction into nuclease or separation of DNA fragments by electrophoresis. See, you cannot separate the fragments before you make the fragments, right? You have to make the fragments first to separate it. And making the fragments is digesting the DNA, correct? Maybe that's why you chose your option, because the difference was right here
Oh,
at the second step.
yes.
So
Mm.
option A is correct because you isolate, then you go to digestion, option the second, then you separate. So 4, 2, 1, and then 6, 3, 5, which is once you separate, then you transfer the separated DNA to a membrane, you hybridize using the probe that you have made, and finally you detect the hybridized DNA. Okay, and if you see same detection in two samples, the two samples belong to the same person. That's how criminal cases are solved by DNA fingerprinting. It's called um, DNA test in normal layman's language. Yeah.
Yeah.
So even if sometimes you're confused, so don't leave these kind of questions, because these kind of questions they give you enough hints themselves. So if you just read, you will realize that okay, first I will isolate the DNA, then separation doesn't make sense. First I have to digest, and then you can keep seeing the options. If you know the first three steps correct, you get the right answer. That's the perk of these kind of questions. So if you know that we first have to isolate, then we have to digest, and then we have to separate, you know that 4, 2, and 1 has to be the starting sequence. And there is only one option like that, 4, 2, 1, which is option A. There is no other option like that. Make sense? So I'm just telling you some tricks to do better in entrances. Yeah? Does it make sense?
Yes, sir.
Yeah. So that's what management is, which I was talking about. Okay, perfect. So you told me that you have to attend
Yes,
another
sir.
class, right? So let's then leave the class here, and we'll continue doing evolution. We'll take questions from evolution and on Wednesday then. So let's keep
Yes.
Wednesday for both evolution and biotechnology. Yeah.
Okay.
Wednesday you will be available full time, like for the whole duration of the class.
Yes, sir.